Let's talk about proteins. Dietary sources of protein, when broken down during digestion, provide the monomer building blocks your cells need to build their own complex protein macromolecules, which look something like this: a complexly folded three-dimensional structure. In this lesson, you will learn four layers of complexity contributing to the three-dimensional shape of proteins. These are fascinating macromolecules because proteins comprise the majority of the dry weight of every cell. They serve as machines performing cellular work and are a component of almost every anatomical structure. Each protein has a unique shape that predicts its unique function in your body. As you study macromolecules, including proteins, lipids, carbohydrates, and nucleic acids, you may notice the repetition of a few key themes. See if you can apply these to proteins. First, a macromolecule's function emerges from its molecular structure. Second, information is encoded in the order of monomers composing the sequence of the macromolecule. And last, since macromolecules are built by cells, their molecular composition depends on the cell's genetics. That is to say, the arrangement of the macromolecule's monomers is genetically determined. Here is a definition of proteins. Recall that each macromolecule is composed of its own type of monomer, and the monomer building blocks of proteins are called amino acids. These computerized images on the right demonstrate how proteins fold like complex origami. Notice that in both examples, collagen and Rubisco, the protein is composed of multiple polypeptide parts. Each polypeptide is encoded by a gene in the cell's DNA. They are individually built by ribosomes. Then these polypeptides come together, chemically bond, and form the final functional protein. Take away any of the component polypeptides and you no longer have a collagen or Rubisco. You probably just have a pile of crap. Useless cellular debris that will need to be cleaned up. Amino acids have two parts. The first part is shared among all amino acids. Stemming off of a central carbon is a nitrogen-containing amino group on one side and a carboxyl group on the other side. The second part is a variable R group that differs between the different types of amino acids. Notice the exciting chemistry on the edges of an amino acid. These bond via dehydration synthesis to each other, forming linear sequences like beads on a string. Here you can see how they align, head to toe if you will, and the resulting carbon to nitrogen bonds, which are polar due to the difference in electronegativity between those atoms, is given a special jargon name. These bonds fusing amino acids together are called peptide bonds. 20 amino acids exist in nature, although others have been crafted by organic chemists in the lab. Notice the shared part of their molecular structure that is the same in all amino acids. Contrast that with the R groups, which are all unique. The R group defines the amino acid's chemical properties, for example, whether it's nonpolar or polar, or positively or negatively charged, and thus its bonding behavior. Now imagine all the possible combinations of these 20 amino acids in a sequence. Polypeptides vary in their length. Some have more or fewer amino acids total, and they vary in the order of amino acids in their linear sequences. If you stirred amino acids together in a bucket, they would bond into polypeptides randomly. But cells can predictably control the order of amino acids, building specific polypeptides on purpose. Until recently, cells were the only thing that could do this, but now humanity has invented technology to build polypeptides outside of cells. Now that we've established that amino acids are the monomer building blocks of proteins, let's imagine a protein's life. It's born from a ribosome that put amino acids in a particular order under the instructions of the cell's DNA. Because all amino acids have the same backbone, these backbones interact with each other to form repeating patterns of folding. If we zoomed in all the way, we could see the string of amino acids twirling into swirligigs and folding like paper fans all along the line. If we took a step back, zooming out one level, we would then see the R groups interacting in a variety of ways. Because each amino acid's R group has its own properties, and each polypeptide has a unique sequence of amino acids, these R group interactions result in a unique, complex, three-dimensional shape that is unique to each polypeptide. But we're not done yet. Remember how collagen and Rubisco contained multiple polypeptides? These already complexly folded individual polypeptides can then combine in various ways to produce many different multi-polypeptide megastructures. Uniting with their teammates in this way produces the final functional form of the protein that we would then recognize with a name. In human bodies, this might be keratin or collagen, or this example, it is hemoglobin, which contains 4 polypeptides, each encoded by a different gene. This is the protein that picks up oxygen in your lungs, carries it throughout your body, and delivers oxygen to your cells. Finally, at the end of a protein's life, there are degrading enzymes that break the bonds, unraveling the protein's folds. Eventually, protease hydrolyzes the peptide bonds, chopping up the polypeptide into individual amino acids. These can then be recycled by a ribosome and get incorporated into a new polypeptide, and the life cycle begins new. Here are jargon words for each of these stages of a protein's life. The order of amino acids is called primary structure, the primary structure of the protein. Next, the repeating patterns of swirligigs and paper fans that result from the backbones of amino acids interacting locally is called the protein's secondary structure. These swirligigs are called alpha helices, and the paper fans are called beta-pleated sheets. Next, the unique fold of an individual polypeptide, predicted by the interactions between its amino acids R groups, is called the tertiary structure. And finally, the mature functional protein, usually composed of multiple polypeptides, is the protein's quaternary structure. If a protein loses its quaternary structure, it is said to be denatured and it no longer functions because a protein's shape enables its function. There are endless possible amino acid sequences and combinations of polypeptides, so there are infinitely many potential proteins. Protein is therefore a versatile macromolecule. However, the real estate on your DNA where genes reside is limited. Remember that one gene encodes one polypeptide. Humans have about 20,000 genes. This is called our genome. But our proteome, the list of proteins we make, is in the billions. For the exam, you should understand exactly how this is possible. Typical human polypeptides are 300 to 500 amino acids long, and no human brain has been able to precisely predict the quaternary structure of a functional protein from its amino acid sequence. However, in the early 2020s, an artificial intelligence model was developed to do just that. This AI now allows scientists to foresee the shape and therefore make good predictions about the function of known amino acid sequences. This is interesting because while quaternary structures denature easily, primary structures, the sequence of amino acids, are recoverable from ancient fossils. This also has great promise for developing pharmaceutical treatments or, more terrifyingly, biological weapons. Here's a brief review of what we just learned about proteins. Amino acids are the monomers of this macromolecule, and 20 amino acids exist in nature, differing only in their R groups. The sequence of amino acids is called the protein's primary structure, and it is formed under the direction of the DNA. Its bonds are catalyzed by ribosomes. And proteins have as many functions as there are ways to fold and combine polypeptides. The final folded form that serves a function in our bodies is called the quaternary structure, and you met 3 example proteins: collagen, Rubisco, and hemoglobin.